So today we're going to dive deep. Literally, we're talking about the deep ocean, one of the last great frontiers on Earth. And hidden down there, scientists are discovering this absolute treasure trove of fungi. We're going to explore the revolutionary method they're using to decode them and, unlock all their secrets. I mean, just think about it. Our planet is mostly ocean, right? And yet it's still one of our least explored frontiers. It's a world of immense pressure, total darkness, and life forms that are just, wow, completely unlike anything we've ever seen. So what amazing discoveries are just waiting for us down there in the deep? Well, it turns out the ocean might be hiding a massive untapped pharmacy. Seriously, a pharmacy. Scientists are starting to believe that the organisms living in these extreme environments could hold the key to developing some seriously powerful new medicines and industrial compounds. And right at the heart of this pharmacy are what are called ocean-derived fungi, or ODFs for short. Now, these organisms have evolved to survive in some of the harshest conditions on Earth. And to do that, they produce these truly unique natural compounds. This isn't just some scientific curiosity. This is a potential gold mine. We're talking about new antibiotics, powerful anticancer agents, maybe even game-changing industrial enzymes. The stakes here, well, they couldn't be higher. Okay, so if these fungi are so promising, what's the holdup? The big challenge lies in what scientists call the non-model problem. And really, it's the central reason why this hidden pharmacy has remained locked away from us for so long. To really get what's going on, you have to understand the difference between two kinds of organisms. On one hand, you have model organisms, like lab mice or E. coli. We know them inside and out. We have all the tools to study them. On the other hand, you've got non-model organisms, like these ocean fungi. They represent the vast majority of life on Earth, but we lack the tools to study them. It's a huge scientific blind spot. So, the crucial point is that this non-model status is a massive roadblock. It's like having a library full of incredible books, written in a language that no one can read. The potential is right there, but it's totally inaccessible. So how do we finally crack the code? The answer lies in this brilliant scientific approach called proteogenomics. I want you to think of it as a powerful marriage between two distinct fields of study, where they combine their strengths to achieve something that neither one could ever do alone. And here is that marriage in a nutshell. Proteogenomics combines genomics, which is the study of an organism's complete DNA, with proteomics, the study of all of its proteins. By bringing these two together, we get a much, much more complete picture of what's actually happening inside the organism. Let's break that down a bit. First, you have genomics. This is all about sequencing the DNA. The best way to think about it is like getting your hands on the complete, unabridged architectural blueprint for a building. It has all the instructions for absolutely everything. Then you have proteomics. So if genomics is the blueprint, proteomics is like going into the finished building and taking a full inventory of every single machine, every worker, and every piece of equipment that's currently up and running. These are the proteins. They're the active machinery that are actually doing all the work that the blueprint described. So, we get the concept of proteogenomics right. Blueprint plus machinery. But what does it look like in a real-world lab? How do scientists actually use this technique to decode a fungus and find those valuable compounds we were talking about? Whoa. Okay, this looks complicated, I know. This flowchart shows the entire process, from a sample of fungus all the way to the final data analysis. But don't worry, we're going to simplify this whole thing into 4 core steps. All right, here's that complex process broken way down. First up, cell disruption. Scientists carefully break open the fungal cells. Then, protein extraction. They isolate all those proteins inside. Next, the proteomic analysis. They use a really cool machine called a mass spectrometer to identify every single one of those proteins. And finally, the big one, the genomic merge. This is where they match those proteins they just identified back to the organism's genetic blueprint. And now we get to the payoff. Why go through all this trouble? This is where we get to see the true power of proteogenomics and how it unlocks nature's most valuable secrets. So the crucial point is this. When you match a specific protein back to the gene that codes for it, you're building a map. You can see exactly which part of the DNA blueprint is responsible for building a specific piece of machinery. This allows scientists to pinpoint the exact genetic assembly line that's producing a valuable compound, like a potential anticancer agent. Think about it. They're not just finding the treasure, they're finding the treasure map. And look, this isn't just a niche interest. As this quote highlights, there's a growing wave of excitement in the scientific community. Researchers are realizing that these non-model organisms, or NMOs, represent a frontier for brand new discoveries. They hold insights that are simply not possible to find in the organisms we already know so well. And that brings us to a final, really exciting thought. Proteogenomics gives us the key to unlock the secrets of ocean fungi. But it doesn't stop there. This tool can be used on countless other non-model organisms all across the planet. So the real question is, what else is out there, just waiting to be discovered?